Tam. I'm a senior at Lowell, and over the summer I worked with Julia Yi in the Blayout Lab in the Regenerative Medicine building, researching the role of IDH1 and 2 in mouse embryonic stem cell differentiation. That's what I was covering. So IDH1 and 2 enable DNA and histone demethylases, leading to sulfate transitions, including differentiation. Mutant IDH1 and 2 occur frequently in blood cancers and brain cancers, and they inhibit DNA and histone demethylases, including TET and Germanic domain proteins. And this seems to contradict what we know in stem cell science, because the promotion of TET and Germanic domain proteins promote reprogramming. So why does the inhibition of these proteins also lead to a less differentiated cell type? To answer this, we looked at the role of IDH1 and 2 in sulfate transitions, focusing on embryonic stem cell differentiation. So the embryonic stem cells we used were called dual reporter cells. Basically, they fluoresced red when they were in an ES cell state, and they fluoresced green when they had differentiated into the epiblast stem cell state. The first experiment we did was to profile the gene expression of IDH1 and 2 in differentiation, and these are some representative images of a control and of a test sample, and these are clearly differentiating because they are losing the characteristic ES cell colony formation, and they are turning green. I measured gene expression levels through qPCR, and our controls behaved as expected. IDH1 and 2 seem to decrease early in ES cell differentiation and then begin to recover after a few days. A second experiment we did, or rather Julia did, was testing the effects of IDH1 and 2 knockdown in ES cell maintenance and in ES cell differentiation. So these are representative images of knockdown controls and IDH1 and 2 knockdown in ES cell maintenance and in ES cell differentiation. So I calculated for some ratios of green fluorescence area to cell area, red fluorescence area to cell area, and green fluorescence to red fluorescence. As expected, the cells that were not differentiated had very little green fluorescence, and the other controls, our controls behaved as expected, and there was no real difference in differentiation or ES cell maintenance between the IDH1 and 2 knockdown samples and the control samples in the same conditions. A third experiment we did was creating IDH overexpression constructs, and sequence results for our IDH1 overexpression construct came out with a match, so we further tested it for blue fluorescence because the backbone we used had a blue fluorescent protein in it. Our controls behaved as expected, and the sample we had transfected with the overexpression vector turned blue. We also measured gene expression levels with qPCR, and our controls behaved as expected, and IDH1 was definitely overexpressed in the sample that we had transfected with the IDH1 overexpression construct. In conclusion, IDH1 and 2 levels decrease early in ES cell differentiation and then begin to recover after a few days. There was no real difference in differentiation or ES cell maintenance between the IDH1 and 2 knockdown wells and the control wells in the same conditions. And our IDH1 overexpression construct works. Possible future directions include making an IDH2 overexpression construct, making mutant constructs, and testing whether the metabolite produced by the mutant IDH1 and 2 mimics the effects of the mutant IDH1 and 2? And if it does the metabolite produced by the wild type reverse the effects? I would like to thank Julia Yi for her mentorship, the Blaylock Lab for having me, UCSF for allowing me to work here, and Mr. Shapiro for taking care of us, and the Lowell Science Research Program for this opportunity to do science research. Thank you.